Hello, my name is Andrew Innes. I'm happy to be here and I'm grateful for this opportunity. So, without further ado, we're going to get right into the lesson. Okay, so what we're going to talk about today is a foundational concept in biology. It's one of the biggest reasons why we eat because it's one of the biggest reasons why we have energy to do anything in our body and throughout our daily lives. We're going to be talking about glycolysis and how it fits into the picture, the more broad picture of oxidative phosphorylation. So, throwing on a few words already. What is glycolysis? So, if we come back here with glyco, this prefix is for glucose, or the six-sided carbon molecule that we'll be talking about soon. And then lysis, or the breakdown of something in general, in this case, that sugar molecule. So, how are we going to learn this today? Through drawing. And why is that? Because most likely, if you can draw out a certain concept, that displays mastery in that content. So, let's start off with a brief overview. Bear with me as I make this initial drawing. So, not the best, but if you can draw it, then roughly, and explain it, then that's good. So we have a person here, and we see a dollar bill sized organ in their abdomen known as the pancreas. So what are we doing this whole process for? In order to get energy. How are we going to get this glucose that we're getting the energy from into our cells in the first place? Well, we're going to eat a burger or pizza or some other food containing carbs, carbohydrates, and break that down in our gastrointestinal tract, absorb it, and then we have to somehow get the sugar from our blood into our cells. And the pancreas is going to release something called insulin into our bloodstream to allow us to pick up those glucose molecules in the first place. All right, let's come back in for a second. All right. So that's a more broad overview of what's happening to glucose. Now we're going to talk about what happens when you actually get in the cell. So here's our, again, all rudimentary drawings are welcome. Here is, we'll label it, glucose. Remember, six-sided carbon molecule. This is a sugar monomer here that we're looking at. So with that, we're going to try and get this into the bloodstream and then into our cells using insulin from the pancreas. And then we're going to break that down through the process of glycolysis. So we're talking about breaking down. What are we trying to break it into? Well, two different things. We're going to talk about pyruvate. And what we ultimately want to achieve to get energy to do these daily processes is ATP. Yes, almost done. And here. So in the process of glycolysis specifically, we're going to break down the six-sided carbon molecule into three carbon molecules known as pyruvate. Those are going to go through another process that will allow us to put this molecule into the Krebs cycle. That's the cycle that precedes that comes after glycolysis in order to continue this molecular breakdown so that we can end up with energy at the end of the day. And that energy is going to be in the form of ATP. So this is a rough drawing of it, but it's adenosine triphosphate. So we have a base here, which is adenosine, with a sugar group, which is the ribose, and then we'll have these three spheres, if you can see that, are the three phosphates in ATP, or adenosine tri for three phosphate. That is ultimately what we're trying to make, and we're getting that from the glucose that we ingested from that pizza or that burger. Know it's a lot of terms going around, but this is all we're gonna piece this all together. So let's look at the cell. where we want to get that glucose in the first place, thanks to our friend the pancreas and insulin. We're just going to focus on one organelle, or two if we're being more specific, since we're talking about the cytosol, as in organelle. So here's the cell, a rough drawing of the cell. We don't have the nucleus here or Golgi apparatus or other familiar organelles, but we do have the mitochondria. Here's the mitochondria, and here is the space within the cell, or the fluid in it, the cytosol, within the cytoplasm. Glycolysis is going to occur in this space. And then when we have two molecules of pyruvate, we're going to have those be transported to the mitochondria for further breakdown. And this is the place, the organelle, where we're ultimately going to get that ATP we want. We're going to zoom in on the mitochondria now for a closer inspection. Glycolysis and oxidative phosphorylation as a whole, there's a lot of different parts to it. And I know it feels like I'm going through it a little fast, but we just want to get a broad overview for this video. So just as a quick recap, again, we eat that food, the hamburger, pizza, and what have you, gets into our bloodstream. We want to get that into our cells. We want to get this glucose into our cells so we can break it down into these different molecules for energy and being able to do things like give this video. And the step in the process that we're in right now is after we've got into the cell, after we've broken down the sugar molecule into the 2-pyruvate in the cytoplasm of the cell, now we want to take those components to the mitochondria of the cell. So now we zoomed in and we're here. So you notice these folds in the mitochondria, right? These are called cristae, and they give more surface area to the membrane of the mitochondria. Why is that important? Because the last step of oxidative phosphorylation and the second step that comes after glycolysis in oxidative phosphorylation is that of the breakdown of the ETC, or rather the breakdown of our preceding molecules in the ETC, or electron transport chain. So the more surface area we have for these ETC, for this electron transport chain to reside on, the more energy we can produce, because there's more area for these to reside. So we'll have more residing in a larger area. So what we have is, after we go through the Krebs cycle, what we're going to do is produce things called NADPH and FADH2. These are electron carriers. So this is what's going to give us the most bang for our buck in terms of producing large amounts of ATP. We get a few in glycolysis because we have the first part of glycolysis where we put into ATP, but then we get out 4 in the end of it. So that's good, but that's not going to be enough to, do things like learning these biological concepts. That takes a lot of brain power. Then we have Krebs cycle. That gives us a few more ATP, but we want to get a large amount. So what we do is use those components that were broken down from glycolysis and put them in the Krebs cycle so that we can get these electron carriers in the form of NADH and FADH2. These are going to go to the electron transport chain in the mitochondria located in the inner membrane. And this is what's going to take these electrons that are at a higher energy and move them down the chain to that at a lower energy. And we're going to use this. We're going to stay broad here, so we're not going to get too specific. We're going to use this in order to run a process that will give us that ATP yield that we're looking for, which is about 32 to 33 or 34 ATP molecules per one molecule of glucose being broken down. So this was a rough overview of glycolysis, sugar breakdown, going into the Krebs cycle and the ETC, as well as the ultimate oxidative phosphorylation. That's that process that we're using those components from the electron transport carriers to make that high yield of ATP. and giving us a kind of a very large view of why we eat and how we get energy from our food. Thank you.